Good afternoon, everyone. Pleasure to welcome you here to the Wednesday afternoon lecture on a beautiful Wednesday afternoon. Glad you're all here to listen to what I think is going to be a very interesting presentation on immunology and some new developments from the speaker who has actually been a leader in this field. Maria Roncarolo is from Italy and currently is the scientific director of the San Rafael Scientific Institute, a position she's recently taken on in which she shared with me presents certain challenges in terms of administrative time commitments, something that I can certainly relate to. But she is continuing to do interesting research, which she will be telling you about. Her research has stretched across many different aspects of human immunology, from basic studies where she is a leader in the demonstration and biological characterization of the first subset of inducible regulatory T cells. But she's also been involved all the way to the clinic and played a significant role in the first gene therapy clinical trial, not the first, but the successful gene therapy clinical trial for SCID due to the lack of ADA, obviously a major positive development in the field of gene therapy. She has received a number of honors that I won't have time to go through. I will say that she's done most of her work in Europe, except for 10 years. She was at DNAX in Palo Alto, but since 1998, she has been at the San Rafael Telethon Institute for Gene Therapy, and as I mentioned, is now the scientific director of the San Rafael Scientific Institute. So, without further ado, let me ask you to help me welcome to the podium Dr. Maria Roncarolo.
Thank you very much, and thank you for being here this afternoon. It is a pleasure and an honor for me to give this lecture. And what I will try to do is to give you in the next 45 minutes an overview of the field of regulatory T cells and our most recent work with inducible TR1 cells. And of course, this is a story that goes back more than 20 years when we started to study patients that were tolerant. And in this patient, we could identify TR1 cells. I like to remind you this because this is an example of how a clinical observation can lead to a basic research project. And then in the last 10 minutes of my talk, I will present actually for the first time the results of our clinical trial, first clinical trial with adoptive cellular therapy with TR1 cells. So I don't need to remind you that in the last 10 years, regulatory T cells have been demonstrated to play a crucial role in inducing and maintaining peripheral tolerance. And we know now that indeed regulatory cells regulate not only immune responses to self-antigens, but also to alloantigens in the context of transplantation and to non-pathogenic antigens, and also to tumor antigens and viral antigens. And I guess I need a pointer because this one doesn't, yeah, perfect. So we know that they are relevant for all these immune responses, but what we do not know yet is whether the same subset of regulatory T cells play a role in each of these responses, or there are multiple subsets with specialized activities. We know very well that the best characterized regulatory cells are comprised in the CD4 positive T cell subset. And among those, we can classify the natural Treg as the Treg which expresses high levels of CD25, FOXP3, and also we know now that they are CD127 low, and they can be CD45 array or RO, depending on the stage of activation. And the other big family are the inducible Treg. And the major difference between these two categories is that the natural Treg are selected in the thymus and emerge from the thymus as a specified population of cells devoted to immunomodulation. On the other hand, the inducible Treg derive from a naive T cells, which sees the antigen in a tolerogenic environment. And among the inducible Treg, I'm going to focus my talk on the TR1 cells, which we described more than 10 years ago, and are defined based on the cytokine production profile. But also, I would like to remind you that there are other subsets of inducible Treg which can play a role in peripheral tolerance. So just to briefly remind you the major biological characteristic of natural Treg and inducible Treg. So the natural Treg are cells, as I told you, that express high level of FOXP3 constitutively. They also have CD25 bright on the surface. These cells are thymic derived. They are anergic in vitro, although we know now that in vivo they can proliferate quite significantly. They can be expanded in vitro, and their expansion is IL-2 dependent. And also we know that they express high level of granzyme A. These cells suppress effector cells through a mechanism which is not yet well defined. We know that you need cell-cell contact between the regulatory cells and the effector cells, and also with the APC. We also know in some preclinical models that cytokines play a role, but we could not demonstrate a direct effect of cytokines in the suppressor mechanism of these cells, at least in vitro. So in vitro, there is probably a signal which is still unknown that mediates the activation, the trigger of these cells, and the suppression of the effector cells, which results in downregulation of proliferation and interferon gamma production. So all the studies that have been performed so far are based on the identification of these cells based on the CD25 and FOXP3 expression. However, I have to remind you that these two markers are not really specific for nTreg, at least in the human system, because these are also markers for activation of effector T cells. And indeed, the only way today in which we can really distinguish natural Treg from activated effector T cells, which express CD25 and FOXP3, is based on the demethylation of the FOXP3. We can identify in this Treg-specific demethylation region an area in which we know that if this part of the gene is demethylated, these are really bona fide nTreg. And when you start to do this type of analysis, you get some surprises. For example, if you look at CD25 positive, FOXP3 positive cells in a patient with autoimmune diseases, you see a very abundant proportion of these cells. But when you look at the demethylation, all these cells are not demethylated, so it means that are effector T cells. So I'm saying this because I think there is a lot of confusion in the field based on the fact that many of us relies on CD25 and FOXP3 as the only markers for bona fide nTreg. So if you look really at this specific demethylation pattern of the FOXP3 locus, you see that indeed the proportion of the real Treg in the peripheral blood is quite low, 1.4% versus the 4-5% that we used to calculate based on FOXP3 and CD25. And if you look also, it's strictly related to age. You see that this proportion of nTreg decreases with age. So what about the inducible TR1 cells? Well, the inducible TR1 cells, I already told you that they are identified based on the cytokine production profile. They produce IL-10, very high level of IL-10, very early upon activation. They produce TGF-beta, but in contrast to Th2 cells, they do not produce IL-4. And in contrast to Th1 cells, they produce very low level of IL-2 and some variable level of interferon gamma. Also, they are distinct from Th17 because they do not produce IL-17. So these cells are induced by IL-10, and I will come back to this concept later on, but basically IL-10 is the major player in the induction of TR1 cells. We also know that these cells, as the natural Treg, are anergic in vitro, but can proliferate in vivo, and we can expand these cells with IL-2 or with IL-15. And indeed, TR1 cells express high levels of the IL-15 receptor alpha chain. So these cells express high level of granzyme B. And indeed, the expression of granzyme B is strictly dependent on the IL-2 and IL-10 production by these cells. But interestingly, in addition to expressing granzyme B, these cells secrete very high level of granzyme B. They suppress through IL-10 and TGF-beta production. So indeed, the suppression mechanism is pretty well sorted out. If you activate the TR1 cells via the T-cell receptor, they secrete IL-10 and TGF-beta. And IL-10 and TGF-beta downregulates the antigen-presenting capacity of dendritic cells or monocytes. They downregulate class II expression and also a number of co-stimulatory molecules and pro-inflammatory cytokines. But also TR1 cells and IL-10 and TGF-beta have a direct effect on the T effector cells because they downregulate IL-2 production and also TNF-alpha production by T effector cells at the transcriptional level. So this is the suppression mechanism of these cells, and when you block IL-10 and TGF-beta production by TR1 cells, you block the suppressor activity. As I told you before, these cells do not express FOXP3 constitutively, and this is a major difference with the natural Treg. However, as Th0 cells, when you activate these cells in vitro or in vivo, they transiently upregulate FOXP3 and also CD25. This upregulation is transient, and quantitatively the expression of FOXP3 is always lower compared to nTreg. But again, if you do a FACT scan, you clearly see FOXP3 expression by activated TR1 cells. And as I told you before, we recently showed that granzyme B is secreted by TR1 cells, and we know now that granzyme B also contributes to the suppressor activity of TR1 cells. Indeed, granzyme B contributes to the bystander suppression mediated by TR1 cells because it induces lysis of the APC. It doesn't lyse the effector cells, but lyses the APC and also APC that are specific that present different antigens. So we think that this is a contribution of the bystander suppression mediated by TR1 cells. So what are the evidence in the literature that this natural Treg and inducible Treg play a role in maintaining peripheral tolerance? There are many examples in the literature that really validated this hypothesis. But in my opinion, what is still very crucial is the observation in the patient and in disease situation. Indeed, in humans, the presence of TR1 cells has been associated with tolerance, and I will give you some examples, whereas the lack of CD25 positive FOXP3 positive cells is associated with systemic autoimmunity. And I think that the best example are patients we call IPEX patients with mutation in the FOXP3 gene. In this patient, there is clearly a systemic autoimmunity which is characterized by severe eczema, severe enteropathy, early onset diabetes in the most severe form. I would like you to appreciate the fact that in this patient, not only you have autoimmunity, but you also have a dysregulation of the Th2 responses. Because in this patient, you have very high level of IgE and you have eczema. So in the most severe form, these patients are dead in the first months of life. In the milder form, you see really recurrency of enteropathy, and you do not have type 1 diabetes. So when we started to investigate this patient, there were reported 14 patients worldwide. And now we have characterized in Europe almost 100 patients, which clearly tells me that so far this genetic disease has been underdiagnosed, and most of the time in the milder form is diagnosed as an autoimmune enteropathy. So in this patient, you have mutation in the FOXP3 gene, and most of the mutations are in the 4K domain, but there are mutations all over the FOXP3 gene. And there is no clear correlation between the genotype and the phenotype. We investigated whether the more severe form correlated to specific mutation, but we couldn't find a linear correlation. We also characterize this patient in terms of the presence of CD25 positive FOXP3 positive cells. And to our surprise, in most of the patients, we could identify these cells in the peripheral blood. So you see, for example, in this patient, you have a population of FOXP3 positive, CD25 positive cells, which is almost comparable to normal donors. Whereas, for example, this patient, there are no FOXP3 positive cells, and it resembles more the Scarfy mouse. So most of the patients have expression of FOXP3, and even in the patient in which we do not detect FOXP3 positive, CD25 positive cells, if we do the demethylation, we see that these patients have indeed natural Treg emerging from the thymus. So there is no deficiency in the generation of these cells in the thymus, despite the FOXP3 mutation. What is really defective in this patient is the function of this nTreg. And indeed, when you look in patients in which there is no FOXP3 protein expression, there is a complete abrogation of the suppressor function in vitro, and this experiment was performed using purified, FACS-sorted Tregs according to the CD25 expression. You see that there is no suppression at all, whereas the suppression of the normal donor CD25 positive cells is quite good. We suppress 80% of the proliferation. In the patient with residual FOXP3 expression, the suppression was impaired, but in some situations, especially when we activate the cells with APC anti-CD3, we could see some residual suppression. So this type of deficiency in the suppressive activity in vitro strictly correlated with the clinical phenotype. The patients with the most severe form had no suppression at all in vitro. The patient with a milder form had some residual level of suppression. So we could conclude from these studies that the majority of the patients had circulating cells, and that the FOXP3 expression did not correlate with the clinical phenotype, but definitely the function of these cells was impaired and correlated with the clinical phenotype. One interesting observation we had recently is that if you look at the carrier mother, of course, FOXP3 deficient patients are male, because the FOXP3 gene is on the X chromosome. If you look at the carrier mothers who are absolutely healthy, the X chromosome allele, the wild-type allele, is only expressed in the CD25 positive cells. So in all the other cells, you have a mixture of the wild-type and the mutated allele of FOXP3. In the population of the nTreg, there is a specific advantage for the cells expressing the wild-type allele. So one question we asked recently was, can we differentiate TR1 cells from these IPEX patients? And this question has a relevant implication in terms of defining whether nTreg and TR1 cells are distinct population of cells which are not dependent from each other. The question is always asked when I give talks, are you sure the TR1 cells which are induced in the periphery are not somehow related to the nTreg? And of course, the in vitro data already told us that you could differentiate TR1 cells without FOXP3 expression. But we wanted really to make sure that there was no relationship between these two types of regulatory T cells. And in order to do that, we identify a patient with a null mutation, so a patient in which there was no FOXP3 expression at all, and we tried to ask the question whether we could polarize TR1 cells, starting from the naive population of T cells present in the peripheral blood of this patient. And the polarization protocol is performed according to what we published before, culturing the cells with IL-10 and interferon alpha. And just to make a long story short, I could show you that in the 3 patients we tested with a null mutation, we could polarize TR1 cells in a way which was comparable to normal donor. And we talk about polarization in terms of IL-10 production and no IL-4 production. So of course the unpolarized cells produce both cytokines, but the polarized cells produce mainly IL-10. And these polarized cells, isolated from the IPEX patient, could suppress in vitro in a way which was comparable to the suppression observed with polarized cells obtained from normal donors. So basically, we could polarize tier 1 cells which were functional. We could also isolate tier 1 cell clones from one of these patients with a null mutation. And you see that in this patient, of course, and in the clone generated from this patient, there was no FOXP3 expression. And despite that, the phenotype of the clones was consistent with the phenotype of tier 1 cells, which produce IL-10, low level of interferon gamma, produce IL-5, no IL-4, and very low level of IL-2. So the conclusion of these studies is that basically tier 1 cell line and cell clones can be generated from peripheral T cells of IPEX patients, and also we could generate FOXP3 null T cell clones which had a bona fide tier 1-like cytokine production profile and could suppress in vitro. So this was the formal demonstration that indeed FOXP3 is dispensable for the differentiation function of tier 1 cells. And we think that these tier 1 cells are not sufficient to really prevent the disease, but we find a high frequency of tier 1 cells in IPEX patients with a mild form of the disease, which is controlled by low doses of immunosuppression. So we still think that these tier 1 cells are not sufficient to prevent the systemic autoimmunity, but somehow later on in the life of this patient can compensate and stabilize the disease. So the other important example on the relevance of regulatory cells in human, and I don't want to go into detail of this because it's all published, is the fact that the presence of tier 1 cells is associated with tolerance. I already told you that the first example, the first discovery actually of tier 1 cells was in a SCID patient transplanted with mismatched stem cells. In this patient, there was a chimera between the host and the donor and full tolerance. And this tolerance was not mediated by clonal deletion of the donor-derived T cells specific for the host HLA antigens, but was mediated by the presence of tier 1 cells specific for the host HLA antigen. So this was the first demonstration, and recently we could confirm this observation of tier 1 cells present in the peripheral blood, which correlate with mixed chimerism and tolerance in thalassaemic patients transplanted with haploidentical bone marrow transplant. Also in this patient, we saw that tolerance and mixed chimerism correspond to the presence of tier 1 cells of donor or host origin specific for the HLA antigen of the donor or the host. And the other example, which I think is very informative, is a patient with celiac disease. If you look at patient with celiac disease in remission, you see that in the gut of this patient, you find tier 1 cells specific for gliadin. These cells are not there in the gut of normal donors or patients with different diseases, are very low frequency in patients with full-blown disease, but when you look at patients in remission, the balance between the Th1 cells and the tier 1 cells is switched in favor of the tier 1. Let's move on. The other demonstration, of course, comes from all the clinical and preclinical studies, which I have no time to really discuss in extent, that show that when you induce tier 1 cells or in vivo, you restore tolerance or you maintain tolerance. And from now on, I'm just going to focus on the tier 1 cells, and I leave the nTreg to other people also from NIH who do a terrific job in characterizing these cells preclinically and also for clinical use. So in terms of tier 1 cells, there are many, many examples. One example I already told you are tier 1 cells induced in vivo specific for gliadin, but there are also many other examples that when you induce tier 1 cells specific for self-antigen, you restore tolerance. In my opinion, one of the most intriguing examples in which TR1 cells present in vivo correlate with tolerance comes from what we call immunotherapy to allergens. So when you desensitize a patient to a certain allergen, for example, DERP2 or bee venom, you see that in the peripheral blood of this patient, the antigen-specific TR1 cells goes up progressively. But of course, there are many other examples of TR1 cells generated in vivo also against infectious antigens or also alloantigens. And again, the preclinical data are even more abundant. There are many studies describing the induction of TR1 cells in vivo which correlate to prevention of pathology or cure of disease. One of the most efficient ways we have to induce TR1 cells in vivo is still giving IL-10 to the mice and to the human being. Unfortunately, the early trials with recombinant IL-10 were not successful because IL-10 is not an immunosuppressive drug. So if you give it alone, doesn't stop the disease, doesn't prevent T-cell activation and effector T-cell function. But when you give it in combination to immunosuppression, and immunosuppression which should not prevent TCR activation and TR1 cells induction, so for example, rapamycin or mycophenolate. So if you give to the mouse rapamycin plus IL-10 or mycophenolate plus IL-10, you induce antigen-specific TR1 cells. Antigen-specific TR1 cells which prevent disease, cure from the pathology, and can transfer tolerance to another mouse. So based on all these data, the adoptive transfer of T regulatory cells became very fashionable. And indeed, there were many preclinical data that really gave a rationale for many of us to go in the clinic with adoptive transfer of TR1 cells. And of course, the preclinical data are listed in these slides, and forget it because you cannot read it, but it just helped me to remind you that the first original description of TR1 cells in the mouse in our Nature paper of '97 was basically the demonstration that when you give TR1 cells specific for ova to a mouse, in which you induce IBD by infusing CD45 RB high cells, you can basically prevent the disease if you feed ova to the mouse. So the ova antigen would activate the TR1 cells and mediate tolerance and suppression of the pathogenic CD45 RB high cells. So based on all these data and on our original data from the SCID patient, we and many others embark in protocols trying to use this adoptive cell therapy to restore tolerance. But it's clear that before going to the clinical trials, we have to admit that there are many, many questions that are still unanswered when we go for cellular therapy with TR1 cells or with natural Treg. So most of the studies that are basically already in the clinic right now with a natural Treg are with polyclonal nTreg. So nTreg with unknown antigen specificity. All the study that we did with the TR1 cells, on the contrary, were with TR1 cells specific for a certain defined antigen. We do not know yet whether it's relevant that they are antigen specific or not. We also do not know yet where these cells go, where do they home, if nTreg home to the tissue and TR1 cells home to a different place, or they go in the same place. And also we do not know exactly whether nTreg are more relevant for the induction of tolerance, whereas TR1 cells are more relevant for the maintenance of tolerance. To try to address this question, we went back to a murine model. And I go through that quite quickly just to give you an idea of what we are trying to address. So in this murine model, this is a murine model of pancreatic islet transplantation, a quite simple model in which we basically induce diabetes in the mice, and then we transplant a purified islet from a donor. And we can use islet from a black 6 donor and use bulb C as a recipient. And in this situation, if we do not treat the mice, we have rejection of the islet, which is transplanted under the kidney capsule approximately in 25 days. If you use a more stringent model in which we give the islet from a bulb C to a black 6, we have a much faster rejection. So we have a model of organ rejection where we can test the efficacy of our TR1 cells in terms of cellular therapy. So in this model, we tried first to give to the mice polyclonally generated TR1 cells. So cells that we generate in vitro by polyclonal activation with anti-CD3 CD28 in the presence of IL-10. In this condition, you can see that basically the population of IL-10 producing cells is around 20% of the bulk population. So if we inject these cells or these cells in the mice, in the transplanted mice, we clearly see that if we use TR1 cells in a situation in which we give islet from the black 6 into the bulb, we have protection, which is not great, but is around 40% protection. But if we give TR1 cells in the opposite situation, when we transplant islet from the bulb C into the black 6, we have no protection at all. So in this model in which we have a faster rejection, if we give the polyclonal TR1 cells, we have no protection at all. What about the antigen-specific TR1 cells? So we can generate antigen-specific TR1 cells by treating mice with rapamycin and IL-10, as I told you before. And we generate around 19% of IL-10 producing cells, which are antigen-specific, because when we activate these cells with the bulb C APC, they produce IL-10, whereas when we activate them with a third party, they do not produce IL-10. So if we use these antigen-specific TR1 cells, which we generate in vivo using rapamycin and IL-10, we ask the question whether they could protect in the more stringent model in which we have fast rejection. And the answer is indeed yes. If we inject these cells 30 days after the islet transplantation, we basically see, so if we generate these cells in the mice treated with rapamycin and IL-10 at day 30 after treatment, and we inject them in the transplanted mice at day -1, just this cell therapy can protect the mice, and we have around 40% protection. And this protection is clearly mediated by IL-10 producing TR1 cells, because if you block IL-10 in vivo by treating the mice with an anti-IL-10 receptor monoclonal antibody, you have 100% rejection. So you clearly have protection by cellular therapy with TR1 cells. So what is the conclusion of this study? Well, the first conclusion is that polyclonal TR1 cells are not more efficient, on the contrary, are less efficient in inducing and maintaining tolerance in this model, and that donor-specific TR1 cells are much more robust in inducing tolerance. And this tolerance is mediated by IL-10. So the other question about the homing gave a very surprising answer, in my opinion. If you generate TR1 cells in vivo by treating the mice with rapamycin and IL-10, and also anti-CD45-RB in this case, and you stop the treatment day 30, you see that you have full long-term protection. If you go and look in these tolerant mice where the TR1 cells are, you find them mostly in the spleen. So in the long-term tolerant mice, the TR1 cells are not in the site of the transplant, are not in the lymph nodes, but they reside in the spleen of the mice. And you see that in the draining lymph nodes, they are basically not there. And in the other organs, the natural Treg are comparable to the TR1 cells. So this is quite interesting, and also tell us that basically, if you look in the long-term tolerant mice, these cells are very robust and long-living. And the same occurs when we do the experiment in the nod mice. If we treat the node mice with rapamycin and IL-10, we can prevent diabetes. And in the long-term tolerant mice, we find the TR1 cells mostly in the spleen. And again, you can see that the TR1 cells in the RAPA and IL-10 treated mice are characterized by high IL-10 production, IL-5 production, TGF-beta production, no IL-4, low level of interferon, and low level of IL-2. What is interesting is that when you treat the mice with rapamycin, you also see an upregulation of the FOXP3 positive cells, mostly in the site of the intracellularly in the pancreas. So just to move to the last part, I want to really summarize by telling you that the working hypothesis we have is that the polyclonal nTreg suppress in the early phase of an adaptive immune response, whereas the antigen-specific TR1 cells are there for the long term. And these antigen-specific TR1 cells reside in the spleen. So what we think is that when you have autoimmunity or transplant rejection, the first wave of cells that try to block the immune response are the natural Treg. But eventually, if you have chronic antigen stimulation, you would generate TR1 cells, which produce IL-10, mediates infectious tolerance and generation of de novo TR1 cells, and are responsible for long-term tolerance. So this is our working hypothesis, and we have different preclinical and clinical models to try to address it. So I have 5 minutes left, right? So in the last 5 minutes, I just want to present to you very briefly our clinical trial. So I told you that the rationale for cell therapy with Treg is the idea that if you restore a balance between the effector cells and the Treg cells, you can cure pathology. And in the human system, we can generate TR1 cells in vitro using different approaches. So far, I showed you the IL-10, recombinant IL-10, but we can also use IL-10 plus interferon alpha. Anogara and her group use vitamin D3 and dexamethasone, anti-CD45 RB-RO antibody, etc. But more recently, we found out that the best way to induce TR1 cells is using a specialized population of dendritic cells, which we call DC10. And we have very hard time to convince the reviewer that these are not macrophages, these are really dendritic cells. These are very unique dendritic cells. First of all, they can be generated in vitro in the presence of IL-4, GM-CSF, and IL-10, but they're also present in the peripheral blood and in the spleen of normal donors. And these cells have a unique phenotype. They express CD14, they express CD16, and they also express CD11C and CD11B. They have also a unique cytokine production profile because they produce IL-10 but not IL-12. And they express high level of ILT4, ILT3, and HLA-G. When you prime T cells with this DC10, you upregulate IL-10 production by T cells. And more importantly, when you prime naive T cells with this DC10, you generate a population of anergic cells which are resistant to re-challenge with the same antigen and that produce IL-10 when re-challenged with the antigen. These anergic cells are suppressor cells. So if you put them in vitro in the presence of naive T cells activated with APC or with anti-CD3, they suppress the proliferation and the cytokine production by naive T cells. And this suppression is really mediated by IL-10 and TGF-beta. So these anergic cells contain TR1 cells which are suppressive. So we use this method to generate TR1 cells to give back to patients in an adoptive cellular therapy protocol. And the trial is now completed. I'm going to show you, of course, this is a phase 1 trial. The first question was the safety. It was a very stringent clinical situation because we gave these cells to patients that receive haploidentical bone marrow transplant. And I don't know if you are familiar with this type of transplant, but basically when you do an haploidentical bone marrow transplant, you have the advantage that you have a family donor, but you have a major disadvantage that if you do not deplete of the T cells, you have little GVH, and if you deplete of the T cells like we do now, we give just purified CD34 positive cells, what happens is that these patients are immunodeficient. Are immunodeficient for many months. And if you try to, to really give T cells in the term of donor lymphocyte infusion, you do not succeed in protecting these patients from infection and from relapse of the tumor. So we decided that if we do a cell transfer of TR1 cells in this setting, we could indeed not only prevent graft-versus-host disease, but maybe favor immune reconstitution and preserve some level of anti-tumor immune response. So this was the aim of the trial, and of course the first aim was safety. And the cells that we gave were, in the first group of patients, we gave TR1 cells generated with recombinant IL-10. In the second group of patients, we gave IL-10 energized donor T cells containing the TR1 generated with host dendritic cells, DC10. So we treated so far, I'm going into the details of the patient, but basically we treated so far 19 patients, and we asked the question whether we could have long-term tolerance with reduced graft-versus-host disease and still some level of protection to viruses. So the trial was sponsored by a EU grant reset. And of course, I already told you about the primary and secondary endpoint, and I'm going to show you the data of the patient. So the patient received, as I told you, mega doses of purified CD34 positive cells with no CD3 positive cells. The CD3 positive cell T cell dose was always equal or less than 10 to the 4th. Otherwise, the patient would have lethal GVH. And the patient were myeloablated prior to the transplant and received the IL-10 DLI at day plus 30 after the transplant, where they had myeloid immune reconstitution. 19 patients were enrolled and 12 patients were treated. Of these 12 patients, 9 received the original protocol with recombinant IL-10 generated TR1 cells, and 3 patients received the TR1 cells generated with DC10. I don't want to go into details. I just want to tell you that we had basically a group of patients that were long-term surviving. I would like to remind you that these patients received haploidentical bone marrow transplant as a kind of, last therapy, third-line therapy after all the conventional therapy failed. So in these 5 patients that we had long-term surviving, we had 1 patient that received a higher dose of TR1 cells, 3 per 10 to the 5th per kilo, that ultimately got GVH, and 4 patients that received 10 to the 5th per kilo of TR1 cells and did not get severe GVH. So these patients are all long-term surviving, no side effect from the infusion. They all are free of tumor, and they are all immunoreconstituted. So of course, the conclusion is that the TR1 cell adoptive immunotherapy is safe, and there are some indication maybe this TR1 cell therapy mediates some level of immune reconstitution without severe GVH and still with some anti-tumor effect of the T cells that we infuse. And this is just the immune reconstitution. I don't think I need to go through this. Just take the story that basically we have a trial where we demonstrate that the TR1 cells can be injected in the patient. These are antigen-specific TR1 cells, and basically we have no chronic GVH, no severe acute GVH, and long-term immune reconstitution. Of course, we are now planning to expand this trial to other indication, bone marrow transplantation in unrelated HLA match, bone marrow transplant donor, and also in thalassemia and in pancreatic islet transplantation. So of course, the adoptive immunotherapy with TR1 cells is one of the many approaches we can have. There are people that are using polyclonal nTreg, that are trying to generate nTreg specific for the antigen by engineering the TCR. There are plans to use TGF-beta induced Tregs. I think that the bottom line is that we hope to have from these cell therapy trials some insight on the efficacy of these cells and also on the mechanism of action. But ultimately, I think that this cell therapy trial should be the foundation for using, drugs that induce these cells in vivo. So I would like to thank my collaborators, Rosa Bacchetta, she is in charge of the IPEX program, Manuela Battaglia, she works in the Diabetes Research Institute, and also very smart PhD student that did the work in the islet transplantation. And this is the team that did the clinical trial with the TR1 cells. Thank you for your attention.
Thank you for a very stimulating presentation. We have time for questions. There are microphones in the aisles, so please, if you have a question, pose it now. Yes, start over here.
You pointed out that you made your first TR1 cells by feeding ovalbumin. And I wonder if you can tell for me, what is the difference when you feed ovalbumin and get tolerance, or you feed polio and get vaccination? What is the difference between oral tolerance and oral vaccination?
Of course, I don't know the answer to that. I can say that in the original experiment, we could demonstrate that these TR1 cells were activated by the ova, and that you need the ova to activate them. These are cells that we give. For example, if you give an antigen in a certain way, you do not get TR1 cells for a long time. If you immunize, let's get away from oral tolerance for a second. Let's go for normal vaccination. If you immunize the kids with tetanus toxoid, you don't get TR1 cells. But if you boost these kids many, many times, like we do quite often after bone marrow transplantation, to restore the memory pool, you get TR1 cells. So the TR1 cells need the antigen to need chronic stimulation to be generated, and need the antigen to be activated. With the polio, I think that, of course, you can say in a situation in which you have no inflammation, you have tolerance. In a situation like the experimental model in which we give the CD45RB cells, you induce inflammation and activation of the immune system. But this is, of course, pure speculation.
Okay.
Over here.
I was interested in your studies on the homing of the TR1 cells in your mouse islet transplant studies, but then you also showed that antigen specificity is important. So did you look in the model where you were able to either transfer antigen specific or non-specific, and do they have similar homing patterns? And if so, then what is the antigen specificity doing? Because you would think that the memory cells, or where the antigen are, where the T cells specific for antigen are going to be in the draining node or at the site. So what are they doing in the spleen? Why do you need antigen specificity if they're in the spleen?
Again, it's a speculation. I think that what I showed you is that when you generate these cells in vivo, so I don't know if when you do the cell transfer, these cells that you transfer would reside long-term in the spleen. So, of course, it was surprising to us that these cells reside in the spleen. And we have now actually confirmed that in the tiger mice, that these cells stay in the spleen. You know, the speculation is, of course, that they can somehow, when they see the antigen-specific cells, or the dendritic cells, they block, they recirculate, and they go to block where they have to be, so that they are stored there, but the action would be when they migrate to the site where the pathology is. Because I don't see how they could see the antigen and block in the spleen. So I just think that the spleen is the storage place.
Simple question. So is there a circumstance that nTreg and iTreg work together in vivo, at least in vitro?
Say again?
So is there a circumstance that nTreg and iTreg cooperate together in vivo, at least in vitro? Could they work together?
So if you give, for example, rapamycin DAL10 in vitro, you see rapamycin, you know, we published already several years ago that rapamycin preserves the pool of natural Treg and somehow suppress the expansion of the effector cells. If you give rapamycin DAL10, you still generate TR1 cells, but they are very few, because they get also suppressed in the expansion by rapamycin, which is not the case in vivo. Because in vivo, when you basically give rapamycin DAL10, you have the expansion of the FOXP3 positive cells and the induction of the TR1. So I'm not sure that the in vitro is the best system.
So do you think whether it's difficult to image both cells in vivo?
To image both cells in
vivo, to
in
see
vivo.
if they cooperate?
That's really the goal. So we are now trying to do that, using the tiger mice of Flavel, in which, you know, the cells that produce IL-10 become GFP positive, and labeling the C25 positive cells. We are not there yet, but we are trying to do that.
Thank you.
Yeah, I have a question. So adoptive transfer of the TR1 or induction in vivo of the expansion of the natural Treg, do you think it might pose a risk for making us more susceptible to infectious disease? Is it a concern, or since the antigen specificity is different? But again, you said that the antigen specificity is an open question, so.
Well, of course, I didn't say openly, but what I was alluding is that when you think to do adoptive cell therapy with polyclonal Treg, whether they are nTreg or a different type, there is always a risk that you have, you know, undesired immunosuppression. With the TR1 cells, the fact that they are antigen specific, it's somehow, you know, a security nest. Of course, there is a contradiction there, because we say that they are antigen specific in terms of that they need the antigen to be activated, but once they are activated, they produce IL-10 TIGIR beta granzyme B, and they suppress effector cells with the same antigen specificity, but also they mediate some level of bystander suppression. So there is a contradiction there. I think that this bystander suppression is very confined. It's confined to the tissue and is not systemic suppression. Every time we look at abundance of TR1 cells in patients, we never see suppression of infection. Abnormal infection, higher frequency of infection. We never see suppression of immune response to vaccination. So I think that if they mediate bystander suppression, it's not systemic. With regard to the nTreg, the polyclonal nTreg, I am very concerned. We have a program also to give nTreg in type 1 diabetes, as many other groups. I have my doubts. Having said that, the preliminary data of the trials in which they gave fresh nTreg to bone marrow transplanted patients, not in vitro expanded, freshly isolated. They claim that they do not have general immunosuppression. There are 2 trials ongoing, one in Italy in Perugia, one in Germany in Heidelberg. They gave this purified fresh population of CD25 bright cells in patients receiving bone marrow transplantation. And they claim that they are safe, that they didn't see more GVHD and they didn't see more infection. But the data are not published yet. So these were just presented at the ASH meeting last December.
Thank you very much.
Well, thank you again for a wonderful presentation. We'll have a reception in the library, so please come if you'd like to speak further with our speaker, and let's thank her again.
Thank you.